Hi, folks. So we've got a couple more non-Mendelian genetics strategies and phenomena that we are going to look at in this rather short video. I imagine this is not going to be around 10 minutes. First, non-Mendelian genetic strategy is going to be pleiotropy. All right, that's what this word is here, is pleiotropy. So when a single gene location affects more than one phenotype, that is considered pleiotropy. So pleiotropy is when you have one gene that's going to affect multiple different features. For example, there is a gene that affects both your brain, your liver, as well as your pancreas. And that's going to be because the same gene, but it's produced in these different locations, and it's transcribed and translated in slightly different ways. So this means maybe it's going to the Golgi apparatus and it's being modified slightly differently. Maybe it's being spliced slightly differently so that it has certain amino acids and slightly different amino acids in the final transcript from the mRNA, right? So there's a variety of different ways that you can get different phenotypes from the same gene. And we learned about a lot of those phenotypes in the molecular genetics unit. What I want you to do is explain how pleiotropy is different from polygenics. Now, if you need to, go back and review what polygenics is, but I want you to take a second to think, okay, what is the difference between pleiotropy and polygenics? You think you have it? Well, the big difference is that they're sort of like the opposite phenomena. With polygenics, if we go back to our example of polygenics is when you have multiple genes affecting the same phenotype. So in this case, we were looking at skin color, but there are multiple genes, A, B, and C. There's actually a lot more, but we just used A, B, and C for this example, that affect the phenotype of skin color. And that's polygenic inheritance. But for pleiotropy, it's the opposite. You have one gene affecting multiple phenotypes. Let's look at an example of this, all right? We're going to combine a lot of information into this example, so I want you guys to be paying attention to ratios and determining phenotypic and genotypic ratios for this example, okay? So a group of scientists bred a yellow mouse with an agouti or a brown mouse. Now, the reason it's not just called a brown mouse is because if you notice, these yellow mice actually have some kind of problematic characteristics. Along with the yellow coat color, they also have things like obesity, they have heart disease, and they have some health issues that are associated with this yellow coat color. And that's this example of pleiotropy, right? You have the yellow coat color, obesity, and all these other phenotypes. And the agouti mice, do not have those same phenotypes. So they're trying to figure out what the inheritance pattern is for this specific gene. Now, their results were a ratio of 102 yellow mice to 99 agouti mice. And they bred a yellow to a brown agouti mice, okay? Now, based on traditional Mendelian genetics, if you assume, which you should always assume starting out, that you are only seeing traditional Mendelian genetics, what type of inheritance might you expect for this gene? And then what are going to be the genotypes of the parental pair based off of this assumption? I'm going to give you a second to think about it. Well, let's think about this for a second, all right? If we were to, I'm just going to quickly give us an extra slide to work with some blank space here. If we were to think about this example, we have a yellow mouse. Oops, that should be yellow. We have a yellow mouse, and we are breeding that yellow mouse with a agouti mouse. And we're trying to figure out what is their genotype and phenotype. Well, if this is based off of traditional Mendelian genetics, meaning there's no codominance, there's no incomplete dominance, there's no epistasis or anything else influencing this particular characteristic, right? We know this isn't the case. We know this is an example of pleiotropy, but you always start with that assumption that it is non-Mendelian genetics, right? So let's say that these are inherited via regular Mendelian genetics. One of these traits has to be dominant and one of these traits has to be recessive, right? So let's just, for example, say that the yellow trait is dominant and then that the agouti trait is going to be recessive. Well, that means the agouti mouse has to be homozygous, all right? You don't necessarily know which one is dominant at this point, but this is just for the sake of getting the problem started, right? So let's go back for a second. Their results in their population were 102 yellow mice, and 99 agouti mice. So this is a ratio. What we've just created here is a ratio of yellow to agouti mice. And what I want you to do is go back to our examples of phenotypic ratios. What phenotypic ratio would typically have a nearly 1-to-1 ratio? Because if we were to reduce this, this is super close to a 1-to-1 ratio. It's not exact. It's never going to be exact. Ratios are never going to be exactly 100 yellow mice to 100 agouti mice, but they're going to be close. And if you take a look at the chi-squared videos and practice problems, how can you tell if it's that close? How can you tell if it's close enough? You do a chi-squared test to figure out if it's close enough, right? Now, this is assuming, right, that one of these is dominant and one of these is recessive. So let's do a Punnett square. Let's say that this individual was homozygous dominant, all right? Let's say that the yellow mouse was homozygous dominant. Well, if we do a Punnett square with these individuals, with a homozygous dominant and a homozygous recessive individual, right? What's the ratio of this Punnett square? Well, if you flesh out this Punnett square, they're all going to be the same possible genotype and phenotype. So this is going to be a 1-to-0 ratio. So that doesn't match up with the ratio that we observed in the offspring. So we know that it cannot be a homozygous individual, right? We know that our yellow mouse cannot be homozygous because that ratio doesn't make sense. So let's go back to a Punnett square. Let's go back to a Punnett square. And now we're going to assume that our yellow mouse is in fact heterozygous. So we're going to say our yellow mouse is heterozygous. Our agouti mouse is homozygous because he has a different characteristic, meaning that he has to have that recessive characteristic. This could also be reversed, but we're running with this example for right now. So this means you would have big A, little a, little a, little a, big A, little a, little a, little a. So does this give us our 1-to-1 ratio of yellow to agouti mice? Well, this would be our yellow mice. And there are 2 out of 4 chances. This would be our agouti mice, which would also be 2 out of 4 chances in our Punnett square, which means this would be a 1-to-1 ratio. So we're going to run with the assumption that this is a heterozygous homozygous cross. Okay? Now, here's where it gets interesting. All right. We got the ratio of phenotypes in the offspring. So this is where it gets interesting, right? So now we're going to pair a yellow mouse from the previous example. So we know that the yellow mice are going to be heterozygous, right? And now that we know they're going to be heterozygous, we are going to breed them together. So what would this Punnett square look like for a cross between 2 heterozygous mice, right? And what is going to be the ratio of phenotypes in the offspring B? What would we expect to see based on regular Mendelian inheritance. Well, if we do this Punnett square and it's regular Mendelian inheritance, what would we expect to see our ratio to be? Well, we would expect to see a 3-to-1 yellow to agouti ratio. Again, because we are assuming that the recessive characteristic is in fact the agouti characteristic. Now, here's where it's interesting. Scientists did not get this result. Instead of getting a 3-to-1 ratio, they got a 2-to-1 ratio in the offspring. They also found that the offspring of this cross did not breed true. Breeding true just means that they were not homozygous. They did not only produce more of their characteristic, right? So the breeding true idea means that if you were to breed a yellow mouse with, or excuse me, a yellow mouse with another yellow mouse, you would continue to get agouti mice, and there was no purity in the yellow characteristic. You never got a yellow mouse that only produced other yellow mice. They always produced either yellow and agouti, and there was never any instance in which they only produced yellow mice. So scientists were looking at this example and they're like, okay, what is going on here? This is really strange. Why are we only producing yellow mice? I want you guys to think about it and think about if you can see what might be going on in this scenario. I'll give you a hint. It has to do with achondroplasia. So if you go and remember achondroplasia, you might be able to figure out this question. Did you figure it out? Well, it turns out that after additional research, these scientists found that these characteristics are experiencing lethality. So in the case where the dominant characteristic is inherited homozygously, the dominant characteristic results in lethality. So that means if you get two dominant traits, that embryo does not survive. So this is an example of pleiotropy, as well as an example of dominant lethality, which achondroplasia is another example of dominant lethality. And this is kind of interesting because if you think about it, these yellow mice, they had a bunch of other characteristics. They had obesity, they had heart trouble, they had diabetes, they had a bunch of other health issues associated with this yellow coat color. And so it would make sense that if you had two copies of those detrimental or deleterious alleles, that would result in an inviable embryo. So it's an interesting example of a lot of different characteristics that we have been talking about, right? This is an example of pleiotropy. And this is how you're able to get a 2-to-1 brown, yellow to brown ratio among the survivors of this generation. Right? So if this characteristic is lethal, why are the yellow mice continuing to survive? Well, I sort of just answered that question, right? The yellow mice are continuing to survive because they have one copy of the non-deleterious allele. They have one copy of the allele that provides them with a somewhat normal physiology, while the other allele provides those health issues in the terms of their physiology, right? So this is an example of pleiotropy. This is also an example of another type of inheritance. It's also an example of another type of inheritance. We said it's an example of dominant lethality. It's also an example of something else. Can you think of what it might be? This is an example of also incomplete dominance. In this case, you have the characteristics of the deleterious allele as well as the beneficial allele blending together, right? So you have some interesting things happening here with codominance and complete dominance. Now, in order to address the next example of non-Mendelian genetics, I want you to answer this question. Do you think that your environment can influence the phenotypic expression of a trait? Now, I'm not asking, can your environment influence how you inherit traits? I'm asking, can your environment influence how that trait is expressed? Use an example. Can you think of any? A couple examples are going to be skin color and hair color. If you're in an environment where you have a lot more sun, your phenotypic expression of your traits might be different than if you are in an environment where there is a lot less sun. For example, blonde hair tends to change color in environments where there is more sunlight. It tends to bleach the hair. And this is true for brown hair as well as blonde hair. But in blonde hair, you tend to really see those characteristics in the specific phenotype. And there's a lot of examples of this. Well, this is an example of the reaction norm. So this is the range of phenotypic possibilities based on environmental influences, right? It's the range of phenotypic possibilities, based on environmental influences. So take a look at this graph. I think I'm too far ahead. Yeah. Take a look at this graph. Let's say that we're looking at anxiety in mammals. So for example, humans are not the only mammals that experience anxiety. Cheetahs also experience anxiety. Now, here's the issue. Cheetahs tend to not experience anxiety when they are in native habitats, or they experience less anxiety when they are in their native habitats. But when they are put in zoo scenarios, they experience really high levels of anxiety to the point where they will not breed. So what zookeepers have done is they've actually introduced Labradors as emotional support animals for those cheetahs in order to reduce their anxiety. So anxiety is not just a human characteristic, it's a mammalian characteristic. So let's use this as an example, right? If we're looking at genotype number 3, so I'm going to circle this genotype, okay? It's the same phenotype, and we're looking at the expression of this phenotype given an environment, right? So what does genotype number 3, what is it indicating about the phenotype of genotype number 3? Think you got it? Well, what this means when we're talking about the reaction norm, we would say that genotype number 3 has a wide norm of reaction. They have a wide range in their expression of a particular genotype, right? It's the same gene, same alleles, but the environment is influencing the expression of that genotype, right? So if we're looking at anxiety in mammals, maybe this phenotype, I'm going to label it in blue, maybe, this particular circumstance is going to be the zoo scenario, right? This is where they have a high level of anxiety. Whereas the green one that I'm going to circle, this would be the low level of anxiety that they experience in a non-zoo scenario. Now let's do the same analysis on genotype number 2. What does genotype number 2 tell us, or what kind of norm would genotype number 2 have? Think you got it? This would be considered a narrow norm. This would be considered a very small range. This would be considered a narrow norm of reaction for genotype number 2. And this is because genotype number 2 does not show a lot of variation depending on the environment. This would be something like eye color, right? Eye color doesn't tend to change a lot depending on the environmental influences. And so you would say it has a narrow norm of reaction. Okay. All right. So that is pleiotropy as well as the reaction norm. Hopefully that was helpful. I'll see you guys next time.